Hello, hello. So welcome to today's session. I'm Sophie Falk, and next to me is Boris Bernhard from McGill University. And we're very excited that you're all here after club night that ended too soon, but soon enough so that everybody's super fresh and listen to these amazing talks. We set the timer at 11 minutes, so you have some room to breathe in case you're prepared for 10. And yeah, we go over to the first speaker.
Yeah, hi everyone. So the first speaker is Olimpia Ortega-Fimbres from UNAM, Mexico. Talking about analysis of focal cortical dysplasias using b-tensor encoding diffusion MRI in an animal model.
Hello, my name is Olimpia. Thank you to the organizing committee, and I would like to show you my work of diffusion MRI for the study of cortical dysplasias. I have no disclosures. Focal cortical dysplasias are malformations of the neurodevelopment that occur in frontal stages. They can vary in severity from slight with normal appearance of neurons to a complete loss of the lamina organization. They have clinical relevance because they are the main cause of drug-resistant epilepsy in children and adults in adults. Also, magnetic resonance imaging is the main tool for the diagnosis and the subsequent surgical intervention. However, many focal cortical dysplasias, such as the 32%, can go undetected. That's why it is necessary to find better methods for their detection. So, if the focal cortical dysplasias can be macroscopically unremarkable, what can be done to dig deeper into the tissue but in a non-invasive way? This is where diffusion MRI comes in, which allows us to have a macroscopic view of what is happening at the tissue level. Diffusion MRI informs us about the organization of the tissue and can characterize the diffusion as anisotropic in elongated structures as axons or isotropic in free water space. But in order to represent the tissue, mathematical models are used. For example, in the DTI model, to represent the white matter who has many homogeneously oriented axons, it is represented by an ellipse. But in the case of the cortex, it is not that the gray matter doesn't have a specific orientation, it is more complex. So the DTI model yields from nearly isotropic diffusion. That's why we wanted to go a little further, examining distributions of tensors, not to derive one or two, but thousands. And we may not be able to visualize so many tensors, but we can talk about their distribution. So here is where multidimensional and b-tensor encoding pop up. MDE bases its sampling not only in its preferred orientation, also its shape. But to better understand this, we have in the first case the conventional way of doing diffusion. But if we have so many things happening within your voxel, you have to ask not one, but several questions. So here is in the left column, the way that diffusion gradients play out to ask water molecules how and where they move. And in the right column is the type of question being asked. So in this project, we use linear, spherical, and planar shapes. And so through this sampling, you can obtain different metrics. From the DTI model, you essentially can get FA, MD, AD, and RD. And on the other hand, thanks to the b-tensor encoding, through the Q-space trajectory imaging, you can obtain new metrics such as CC, which is sensitive to the orientational coherence, micro-FA, which is sensitive to the microstructure of the analyzed tissue, KI, which reveals the heterogeneity of the diffusivities, and KA, which relates to the ensembles of the anisotropic compartments. So for the experimental strategy of this project, was based in four main parts. A murine model of cortical dysplasia, where we use Sprague-Dawley rats from 120 postnatal days, and then all the rats were scanned in vivo in 7 Tesla scanner, considering 3 b-tensor shapes and 4 magnitude of b-values. Also, MVP and GFAP immunofluorescence were done, and we carry out a special statistical analysis, but I will explain it next. So the images were denied and were corrected for geometric distortions, and also we used the QTI plus model to obtain maps from the A metrics that I already mentioned, and then we use a curvilinear coordinate system to sample the dMRI parameters, thus providing anatomical correspondence between the animals. Also, we obtained the metrics of the QTI plus at each point, and here you can see the average from the control and the BCNU group. Also, we evaluate the difference between groups at each point with t-test, and we express the magnitude of that difference with coherence D. From now on, I'm going to show you my results like this. So, among the results from the DTI model, the greatest difference corresponded to FA. The white circles represent statistical significance at cluster level, and you can see here a reduction in the BCNU group in the both hemispheres of the FA. Also, we saw reductions in the BCNU group in MD and AD, being more evident in the axial diffusivity. RD was mostly not different between groups. So regarding the results from the QTI plus model, the greatest differences corresponding to micro-FA and KA, which show reductions in the BCNU group throughout all the cortical layers in the both hemispheres. Also, CC present interspersed anomalies at different depths, and KI show increases in the BCNU group. So at this point, we wanted to know what histological features may drive to the diffusion changes that we saw. So the texture analysis of the myelin basic protein showed differences in the control in the BCNU group, where a notable decrease of the BCNU group was observed in the coherence maps, where darker areas from the medial to lateral area of the cortex was observed. Also, this was corroborated with vectors maps, where swirls of vectors interspersed in the somatosensory area of the cortex were observed, corresponding to a disorganization of the myelin fibers. Also, we evaluate the glial cells through the immunofluorescence of GFAP. This is an example of the control cortex with its corresponding automatic threshold on the right, and this analysis corresponding to M1 motor cortex. So, qualitatively, there are more glial cells, especially in layers 4 to 6, and quantitatively, there are a larger proportion of area occupied by the glial cells in the BCNU group. So, the conclusions of this project were this study demonstrates the usefulness and sensitivity of QTI plus metrics to evaluate macrostructural changes induced by BCNU treatment. Also, the findings that suggest that micro-FA and KA are particularly sensitive to neural integrity and fiber orientation, while CC and KI provide information about the surrounding environment, and those results have implications for understanding the mechanisms of BCNU and for developing potential therapeutic strategies. And also, since b-tensor encoding is possible in humans, our results indicate that advanced diffusion MRI has a lot of potential for the detection of focal cortical dysplasias at a clinical level. So finally, I want to thank the members of my lab, the members of my thesis committee, and the people involved in the technical assistance that I have needed. Thank you so much. Any questions?
So thank you very much. So we have some time for questions. You can either use the app to post your questions, or you can come to the microphone and ask a question, like this gentleman over here.
Hi, very interesting, thank you.
Thank
I
you.
just wondered, why did you use a generalized model? It's a focal cortical dysplasia. There are other models like MAM pilocarpine, Friese model, and so on, and then you could compare hemispheres. Here you're stuck to group comparisons.
We use, for example, the grid lines that those providing anatomical correspondence between the animals, and we thought that the distributions of tensors will be useful for the detection of the abnormalities, and the model showed that there are abnormalities in the BCNU group. So we think that the model that we use is useful for the model of cortical dysplasia that we use.
Thanks.
There's also one question from the poll, and that relates to the visibility of the lesions on structural MRI sequences, and maybe like a follow-up question to that could be whether other quantitative MRI contrasts are also useful to characterize myelin alterations.
I'm sorry. Can you repeat the question?
So the first question would be whether the lesions were visible on structural MRI contrasts, such as T1-weighted imaging or FLAIR.
Okay. We saw that structural images present abnormalities in the BCNU group, but the abnormalities are more like, well, structural. But we wanted to know what histological features are presented in the model that we use. So the model that we use, the QTL plus model, show difference between the groups, but not also anatomical, since also in the metrics. So it's more like sensitive with metrics than the structural images.
Thanks. Final question over there.
Yeah, really great talk, and thanks for that.
Thank you.
I hope I didn't miss it, but what was the sequence you used? What was the resolution, and can we achieve this with clinical sequence as well, or is this a preclinical sequence, the MRI sequence?
If I understand well, what type of sequence that we use?
Yeah, the diffusion MRI sequence you used for the...
Yeah, well, we use the QTL plus model, and then we considering that 3 b-tensor shapes, the linear, spherical, and planar, and also for magnitude of v-values, 200, 700, 1,400, and 2,000. So.
Would you know the voxel resolution? Like the resolution of the voxels, how small they were?
Yeah, we use, I don't remember well, but I can tell you, at the final of the session, I can tell you.
Okay. Thank you, great talk.
Thank you.
Thank you again.
Thank you.